Hello students, welcome to the second lecture of week 1. In our previous class, we have started our discussion about the plant cell structure, and we have discussed some of its basic components like cell walls, plasma membrane, then vacuoles, that all we have discussed. Further continuing, we will discuss many more other organelles which are present in a plant cell. Now the next important organelle is plastid. Now plastids are a very closely related but a diverse group of organelles which are particularly found in plant cells. Now these plastids serve various functions. Okay, that all we will discuss, and the different types of plastids are there, but basically we have identified and we have classified those plastids based on their colors. You have leucoplast, which are basically colorless plastid. We call them as leucoplast. However, different functions are assigned to those leucoplast, and they could be further classified into other groups. Then colored plastids are known as chromoplast, and then we have green color plastid, which is known as chloroplast. Now we generally study lot more about chloroplast, and which are the most important plastid present in green plants and serve very important function which are unique to green plants, that is photosynthesis, and overall the plastids serve as a function of synthesis as well as storage. Now along with that, there are many other types of plastid also, that all we will try to understand in this lecture. Now first, the important plastid is one which is known as proplastid, and actually this name is given because it is considered to be the progenitor of all the plastid. That means all the plastids have been evolved or would have been formed from this proplastid, and this proplastid is found in meristematic tissue, that is dividing tissue, and in those tissues which have attained their permanent structure and function, this proplastid is not present. However, its more differentiated form is present. This proplastid is found in definitely dividing cells which are relatively non-differentiated, and they have very few internal membranes. So this proplastid will also not contain an elaborate system of membranes which is particularly found in chloroplast. So it is found in young and undifferentiated cells. Now coming to the most important plastid which we have studied is the chloroplast. Now chloroplast is a green color plastid because it contains a green color pigment that is known as chlorophyll. So it is a chlorophyll containing organelle which is found in green parts of the plant, particularly in leaves of a plant and green stems also. This chloroplast is present. Okay, and this chloroplast, if we try to observe it under simple light microscope, we can observe them and they appear in the form of green dots, as we can see here. The picture taken under simple microscope, green dots of plastid we can see in cell. Now the number of these chloroplasts can vary from one to many, and depending on the activity of the plant, and their size is also variable, about 1 to 10 micrometer in size. So it is relatively slightly bigger organelle because it could be visible when we try to observe it under microscope. Now the shape of this plastid is basically spherical or oval or disc shape when we observe it under higher plants. However, in lower plants like in different algae, different shape of this chloroplast is also present, like we can observe spiral shaped chloroplast, cup shape, then net shape or spindle shape, which is observed in lower plants. Now the important function of this chloroplast is photosynthesis. We will slightly discuss about it later. Now when we will observe the structure of this chloroplast, we can see that it is a double membranous structure. That means it is bounded by two membranes, the outer membrane and the inner membrane, and in between you have an intermembrane space is also there. Okay, so that means it contains 2 lipid bilayers. So basically 4 lipid layers are there. So 2 lipid bilayers which are separated by a space also. Inside this chloroplast, you can see a fluid filled matrix which is known as stroma. And this stroma is rich in many various metabolites. It contains some ions, it contains various enzymes which are required in the process of photosynthesis. It contains a lot of protein also. And when we observe this stroma, we can see structure, a membranous structure is present in this stroma. And this membranous structure is known as thylakoids. Now, membranous structure means these structures are also bounded by a membrane, and the composition of the membrane is similar as that which is found on plasma membrane as well as other organelles, like in chloroplast also, the outer and inner membrane, they are all made up of a lipid bilayer. Similarly, the membrane which this thylakoid has is also made up of lipid bilayer, but in this case they are made up of single membrane as compared to the this outer boundary of the chloroplast, which is double membrane. Now, these membranous structures are present distributed in this stroma, and if we observe these thylakoid or disc-like membranes are stocked or piled one above the other. They are piled one above the other, okay, and this pilation, this stock is known as grana. Here also we can see, so this is grana, singular is granum. Okay, so this stalk is grana and single lamellae we call it as thylakoid. We can also see that these granas are also interconnected among itself, again with membranous structure. Okay, the composition is almost similar because it is a membranous structure. Now on that basis, we can differentiate two types of thylakoid, that is granule thylakoid, means the membranous thylakoid which are stalked one above the other is known as granule thylakoid, or granule lamellae, means lamellae also means a membranous structure, and around 10 to 100 grana per chloroplast could be visible. Similarly, we have stromal thylakoid or stromal lamellae, that is the one which is interconnecting those grana. Okay. Now, the chloroplast contains its own DNA and ribosome, because of which it can also synthesize some of its proteins, some of its enzymes, some of its RNA. So, these organelles will also be present in the stroma. Along with that, we can also see some lipid globules. Okay, so lipid storage is also there, and some starch granules also we can observe. So these synthesis of starch and storage of starch also takes place in chloroplast. Synthesis, definitely we all know that through the process of photosynthesis, and carbohydrates are synthesized, and a large carbohydrate in the form of starch, they are converted and they are stored, and lipid are also present in the form of globules. Now, in this picture also, which is a electron microscopic image, in this also we can see that lot of grana is there and they are interconnected by stroma lamellae. Now, let us try to understand in detail the structure of these thylakoid and the function which this chloroplast serves for the plant, as well as this the function, that is photosynthesis, is also not only important for plant but for every living organism on this planet is actually dependent. Most of the organism, it's not that every living organism, but most of the living organism depend on the process of photosynthesis for their energy intake. Now, let's try to understand here that what is happening in this thylakoid membrane. Now, suppose this is the chloroplast, okay, containing a double membrane structure, and these are the thylakoids. When we talk about individual thylakoid, let's take it as a membrane bound organelle. So in this thylakoid also it will have a membrane and it will have a lumen inside. Okay, you can say that it's a cavity inside this membrane. It bounds a cavity which is not exactly hollow. It contains various types of chemicals also. Now this membrane is made up of lipid bilayers. So this also you can imagine that two lipid bilayers are present. So in this way, this entire thylakoid will cover a cavity here. Okay. Now in the chloroplast, basically it performs the function of photosynthesis. And we all know that photosynthesis, actually we can divide them into two processes. One is the light reaction and the other is the dark reaction. Light reaction means that it requires the presence of light, whereas dark reaction means that it doesn't require the presence of light. That is, it is light independent reaction. Now, light reaction takes place in thylakoids, whereas dark reaction takes place in stroma. Now, why these reactions takes place in these different parts of this chloroplast? It is only because they contain the necessary components which are required for that process. Now, I am not going to into detail that what is the exact process, but I will try to explain you the organization of those different components, means the structure or means how they are oriented in the chloroplast, so that you can have a better understanding of the structure. Now, stroma also contain a lot of enzymes which are required in dark reaction. Now, in thylakoids, you can see that this light reaction takes place. So, it is a double membrane, it is a lipid bilayer structure. Okay, and this, as in plasma membrane also we have studied that lot of other components are there, basically proteins are there. So in this also lot of other components are embedded which take part in light reaction, like you know that photosystems are there which are responsible for photosynthesis. So in this for thylakoid membrane, these photosystem, basically photosystem is a pigment and protein complex. Okay. So you will find different photosystem 2 will be there, photosystem 1 will be there, then you will have ATP synthase. Okay. Okay. So and along with that you will have other protein components, you will have plastoquinone, plastocyanin. So all these molecules will be embedded in this thylakoid membrane and they will help in transfer of electron. So basically the chlorophyll, actually we talk about chlorophyll, which is the green color pigment which is found in chloroplast, and they are embedded in the form of protein and pigment complex. You will see chlorophyll in these complex and they are forming antenna complex. Okay. So where actually the role of chlorophyll is to capture light. Okay. So the energy which is present in this light is captured by chlorophyll. Okay, and this energy, now with the help of this energy, the chlorophyll molecule will be excited, they will release electron, and these electrons will be carried by from one electron acceptor to another electron acceptor, and through this while doing this process, protons will be pumped inside this lumen, and this potential gradient will be developed, and this proton will move through this enzyme, that is ATP synthase, and this enzyme will finally, with the energy which is stored in the form of chemical potential, they will generate ATP molecules. And here also photolysis of water will also take place in the presence of light. Okay. So we can say here that in photosynthesis, light energy will be actually converted into chemical energy. So in this light reaction, these components will be arranged as such in the thylakoid membrane, and they will help in creating a chemical form of energy, that is ATP. Along with that, another molecule, that is NADPH, will also be synthesized, and these two molecules will again be used up in the dark reaction to synthesize sugars, basically a triose phosphate, a 3-carbon sugar, okay, utilizing CO2, okay. They will take up CO2 and in the presence of water molecules also, and using ATP and NADPH, carbon dioxide will be reduced to a, first of all they will be reduced to a 3-carbon compound, and later many other carbohydrates and many other molecules will also be synthesized. So because of its arrangement, this type of process is possible in chloroplast. Now again coming back, now how this chloroplast would actually is generated. As I have told you that all the plastids are generated by proplastids, okay, because and they are present in the non-dividing undifferentiated meristematic cells. So chloroplast is also coming out from this proplastid. So in this proplastid, we can say that basically it has a very simple structure and small, it is also made up of double membrane. Now the inner membrane forms small vesicle-like structures, okay, and these vesicles, when they are destined to form chloroplast, they will unite together, okay, and they will unite together to form thylakoids, to form a flat disc-like structure. Later these disc-like structure will stack on one above the other to give rise to grana. So in this way chloroplast will be generated. Chloroplast could also be generated by division, by normal means from the pre-existing organelle can also divide and give rise to new chloroplast. Now it is also believed that how the early chloroplasts would have been evolved, and it was thought that some early primitive cyanobacteria, basically which are autotrophs, they would have been performing photosynthesis that were engulfed by a eukaryotic heterotroph, and initially in the beginning they started living together as a symbiont, and later they became a part of that eukaryotic cell and gave rise to a future plant cell. And because of this theory is supported in that way that chloroplast also contain its own DNA, and it also contains its ribosome. Now the ribosome which is present in chloroplast is 70S type, okay, so which is peculiar to prokaryotic organism, and it has, we can say that it is semi-autonomous. It can perform some of its own function, but later the eukaryotic cell component of that partner took over the chloroplast, and most of its DNA was degraded, most of the DNA was lost, and many of the things which is required in chloroplast is actually synthesized from the nuclear DNA. Now coming on to the next important chloroplast, which is leucoplast. Now they are basically meant for storage. Now in that they can store a lot of substances. When starch is stored, they can store starch, so it is known as amyloplast. They store lipid, okay, or oils, we can say, or oils also, so that is known as Etioplast. Or they can store proteins, which is known as proteinoplast or proteoplast or alluroplast. Now when we talk about amyloplast, so definitely it won't be present in much abundance in green plants because they do not contain chlorophyll. They are basically meant for starch storage. Now starch is stored by most of the plastid, even chloroplast also stores starch, but for long term storage the chloroplast or the proplastid gets converted into amyloplast. So this amyloplast is found in basically sink tissues or those tissues which are meant for storage, like fruits, seeds, tubers, okay. So in all those parts of the plant we can find amyloplast. In roots also it is present. However, in low frequency we can find them in various other organelles like leaf, stem and root, but a short-term storage of starch. Similarly, Etioplast, it stores lipid as well as it synthesizes lipid. Okay, and you will find them in those organelles which are engaged in lipid synthesis, like in some secretory glands which secrete aromatic compounds which are basically lipid in their origin, like citrus fruit. Okay, the aroma or the fragrance or the taste which comes out from the citrus fruit is because of its secretory gland contains lot of amyloplasts which stores all those oils and aromatic compounds. Similarly, it is found in tapetum also. And what is tapetum? See, basically, you know, in anthers, okay, anthers are the reproductive organs which produce pollen grains, okay. Anther produce pollen grains. Now, these pollen grains are present in a sac-like structure which is known as microsporangia, okay. Just in simple way how to trying to tell you what is tapetum, not going in any detail of it. These pollen grains, when you will cut the transverse section of this anther, you will see microsporangia. In this microsporangia, pollen grains will be there. Now, this microsporangia is surrounded by various wall layers, in which the lower wall layer is known as tapetum. Okay. Now, this tapetum is very important because it provides food for the developing pollen grain. So, in this tapetum also this Etioplast is observed. Similarly, proteinoplast, it is not very specific, but yes, the organs depending on the requirement of an organelle and at different stages for the requirement of a protein, this proteinoplast could be present. Now, coming to chromoplast. Now, chromoplasts are basically colored plastids, and they are found in organs which are colored, like you will find them on petals, on fruits, even aging leaves also you can find chromoplasts, as you would have seen that when leaves are dying, their green color is removed and they start giving out colors like maple trees there. So, it is because of the conversion of or the formation of chloroplast. So, basically it gives bright red color, orange color and yellow color, and these colors are because of the presence of two very important pigments in chloroplast. They are beta carotenes and xanthophyll. So, they provide color. Now, these carotenes are actually, you would have heard these carotene in terms of photosynthesis, in terms of those chloroplast also, because along with chlorophyll, you have some accessory pigments, and these are the accessory pigments which are also present in chloroplast, but these pigments are actually masked by chlorophyll and you don't see in them. But in chloroplast, you don't find chlorophyll, you find beta carotene and xanthophyll. Now, beta carotene contain hydrogen and carbon, whereas xanthophyll contain carbon, hydrogen as well as oxygen. So, these pigments actually they are accumulated in the chromoplast in the form of pigment protein droplets. That is, pigment is basically lipophilic in nature, and proteins they form a droplet, and this is known as plastoglobuli. So, in this complex you can see in this figure also large pigments droplets are present in this chromoplast. Now, what is the purpose? See, definitely the color things will attract. So, this chromoplast will help to attract insect for pollination and for seed dispersal, like insects will be attracted towards the fruit, they will eat it and they will help in dispersal of the seed. Similarly, the colors of the flowers could be used to attract insect for pollination. In storage organs like carrots and sweet potatoes, which you see that they are colored compounds, chromoplast are also present in these modified roots. Then coming to Etioplast, it is also an intermediate type of plastid between Proplastid and chloroplast. Now the chloroplast, you can say that if proplastid is not exposed to light, they will be converted into etioplast, and if we expose chloroplast, this etioplast to light, then they will be converted into chloroplast. Now it is definitely found in stem and leaf tissue of flowering plants which grow under complete darkness or under extremely low light condition. Now the next type of plastid is gerontoplast. This is also chloroplast derived plastid which is found in senescing leaves or the leaves which are under stress condition or the dying leaves. Now what happens when the leaf is dying? The plant actually degrades the chloroplast, it degrades the cellular components of the leaf and use them for nutrient recycling for other growing parts. So under that process, this chlorophyll molecule, the thylakoid structure which is present in the chloroplast, they all are degraded and they form large lipid and pigment complexes in the form of plastoglobulin, and this degrading chloroplast is actually known as gerontoplast. Now you can see here, this is an electron micrographic picture where we can see droplets of this pigment and protein complexes in this gerontoplast. Now basically all plastids are developmentally related. So we have learned that all plastids arrive from protoplast, okay, now which is a double membrane structure having vesicles, okay. Now these can be converted, this will be converted into chloroplast having thylakoids and grana, okay. In aging leaves, they will be converted into gerontoplast, okay. If in fruit, suppose this chloroplast will be developed into chromoplast, okay. This chromoplast will not have a laminar structure and basically it will have the pigments accumulated in the matrix. Then protoplast could also be converted into leucoplast. Then you can have a amyloplast in which you can have large storage granules, okay. Then you have proteinoplast and then you have elioplast, which you can find in pollen grains, okay. Find in pollen grains, okay. Elioplast, and then if it is exposed to darkness, this protoplast will be converted into etioplast, and if it is light is given to etioplast, then it will be converted into chloroplast. So basically all plastids are developmentally related and they could be converted into other forms of plastid depending on the requirement of the plant. So for today I will end my lecture here. Many of the other organelles are there which we will cover in our next lecture. Now for today's work, I just want you all to just try to recall the things which we have discussed in our two lectures and try to answer these questions or try to find the answer of these questions. The first question which I would like you all to think about is to find out the function of the membrane proteins which we have studied. Now membrane proteins are found on plasma membrane also, chloroplast also. So on general you try to find out the answer that what is the function of membrane protein, and also try to find out the difference between sun and shade chloroplast, okay. Now it is not very difficult, you can easily think of that See, it is not Etioplast. The plant is not completely growing under darkness. You can see that a very big plant on the top, they are receiving complete light, and on the bottom the light is slightly lower. So what could be the difference in the chloroplast of these two parts, so that they can have full they can capture full energy of the sunlight, and you utilize them to their maximum. Okay, so for today, try to answer these questions, try to think about it, and so I end it here. Thank you.